Okay, so welcome again, everyone in the room and online. Those of you who have joined quite recently to the seminar, I'm about to present the 4th of 6 presentations in the area of genomics and health economics. The format is presentations with a few clarifying questions after each presentation, and then we'll have a Q&A session at the end. So my presentation more or less wraps up the area of rare disease. So there are several talks that have been in the area of rare disease and health economics. And my presentation talks about some emerging themes that we're working on here in Oxford and economists are also working on elsewhere. So it's quite a high level presentation. I cover 2 areas, genomic therapeutics and newborn screening. So in terms of advanced therapeutics, we're thinking here largely about developing therapies for many rare genetic disorders. Most notably, CRISPR technologies have been highlighted in the media as really expensive but curative technologies. And these technologies can be beneficial for a range of conditions, including immunodeficiencies, retinal blindness, neurodevelopmental disorders, and brain disease. So the three big areas where these technologies are being looked at the moment are brain, blood, and eye diseases. So what does this mean for us as health economics researchers? What's happening in the space currently? So I use an example here of the CRISPR gene editing technology. It's probably the most widely recognised technology in the area. And I use an example here of therapeutic Casgevy, which uses CRISPR gene editing to target and edit variant forms of genes in the bone marrow, stem cells associated with 2 blood disorders, thalassemia and sickle cell disease. So the big heading is that these treatments can be curative. So unlike other areas of health economics, where we're often talking about managing symptoms or treatments, we're talking about actually curing disease here. So that's the big clinical picture. Now, in terms of this particular therapy, the evidence on effectiveness from a single-arm trial, which is interesting to note, single-arm trial, was given to 44 patients suffering from sickle cell disease in one study, with a median follow-up of just over 19 months. In 30 patients who had sufficient follow-up data to be evaluated, 29 were free of so-called vascular occlusive crises, which are very, very severe side effects of the condition, for at least 12 months. So this suggests that actually it was curative, but obviously there's not long-term clinical evidence at all. And the list price for this particular therapy in the UK is over $1.5 million, and in the US $2.2 million US. So in terms of the regulation and reimbursement aspects of this particular therapy, this is a really interesting test case. So the therapy was developed by Vertex in collaboration with CRISPR, and last year was given approval by the UK MHRA for both thalassemia and sickle cell, closely followed by the FDA approval. And then afterwards, when it came to the reimbursement aspect, NICE reviewed both submissions in the company and gave recommendations to just one of them, only thalassemia. Sickle cell was rejected in this context. If you go to the NICE website, you can Google NICE and the drug, and you'll find minutes from previous meetings. It's really helpful to go through the process and see the various submissions for the 2 different indications for this drug. And you see that for sickle cell disease, it's being resubmitted, and there's various iterations required. So it's not been a smooth process at all from MHRA approval to getting NICE approval. And in terms of health economics in this area, there are several things that we need to think about. Dealing with small sample sizes will be a common problem because these conditions are rare and doesn't include many people. Deciding which health and non-health outcomes to assess, which measurements to use. Considering applying an inflation factor to QALYs, using a higher cost effectiveness threshold, given that some of these technologies are very expensive, but yet could be curative, and we're not very good at looking at the very long-term implications of indications, or perhaps using other forms of economic valuation, such as cost-benefit analysis. So there are lots and lots of things here at play, and the various health economics work in this area is still very at early stages. Okay, so that's where we are in terms of genomic therapeutics. It's a really emerging area of health economists. There's lots and lots of work to be done, literally decades worth of work for us to look at in the future, with newer technologies coming online, more indications. And the hope is that actually over time, gene editing techniques could be used more in common diseases and not necessarily just rare diseases if we're looking at curative intent. Now I'll move on to another emerging area of interest to us in Oxford in terms of health economics and genomics. And this is moving from sequencing for patients, which both John and Sally presented on, to healthy individuals, in this case, newborns. So those of you who keep an eye on the press, you've seen last week the launch of the newborn genomes programme from Genomics England, so-called Generation Study. And the press highlighted that the first newborn babies are started tested for over 200 genetic conditions in a world-leading study in NHS hospitals. The background to this is that currently in the UK, babies are tested for 9 different conditions using a blood spot test, which is a biochemical test, and if something of interest is found, then a genetic test does follow. But we aren't currently using whole genome sequencing in this way in the NHS for newborns. So I said this study has started to test babies for over 200 rare genetic conditions. So we're going from 9 conditions to over 200. And the aim is to screen up to 100,000 newborns in England, with ambitions to increase this in the future to 200,000 newborns in this new study. So the Generation Study is a collaboration between Genomics England and NHS England. And we'll see the babies having whole genome sequencing using blood samples, usually taken from the umbilical cord after the baby is born. But the consent process, etc., is done prior to the birth with the pregnant mothers. And one of the main factors in these 200 selected conditions is they can be treated in the NHS in early childhood. So there needs to be clinical treatment available. So it's not speculative testing of genes, there needs to be treatment. And these treatments can range from metabolic treatments, glucose treatments, through to transplantation. So there's a whole range of different curative treatments for some of these babies. Obviously, most of the babies will not have a condition. That's an important aspect to find out. For those who do have a condition identified, they'll get their results within 28 days. So that's the intent. And those with no condition suspected, it will be within months. So the priority given to those with suspected condition using the whole genome sequencing method. And if a condition is identified as having a treatable childhood condition through using whole genome sequencing, families and carers will be given further NHS testing to confirm the diagnosis and ongoing support and treatment from the NHS. So obviously this implies that the NHS has to be up to speed and prepared for these results being filtered through. So in terms of points of interest here for us as health economists, there are lots and lots of things to consider. First being the impact on primary and secondary healthcare resource use. So the sequencing is being done at scale, but obviously taking blood sample is done in a hospital setting. There'll be other downstream consequences of having the testing, which the NHS will need to consider in terms of resources being available when necessary. And we need to look at what the actual changes are to health-related outcomes and non-health outcomes. So does having the whole genome sequencing test and then a subsequent diagnosis, is it meaningful in terms of mortality, etc., and quality of life? So bear in mind that there does need to be treatment available. Does it actually change health outcomes and potentially health outcomes for the baby and family members? What the uptake of newborn screening using whole genome sequencing will be, no one really knows at the moment. The study is very much in its early stages. It's hopeful that the majority of people who are off date will agree to take part. And then another point to think about is the effect on equality of access to further tests and treatments from the whole genome sequencing. So if clinical tests and treatments are meant to be available afterwards, what does that look like in reality? Are there concerns about equality of access that can't be ironed out quickly? And finally, one important question to ask is why use whole genome sequencing, which, as Fred described, as a very wide sequencing approach. Why is this being used rather than other methods such as whole exome sequencing and panel sequencing, for example? So that's where we are in terms of some emerging thoughts here in Oxford. We welcome collaboration with people out there because we're aware that not many people are working at the moment on these two areas, particularly the genomic therapeutics. So we're really keen people to collaborate with us. And just a final mention that I'm partly funded by the Oxford NHR Biomedical Research Centre. I'm very grateful for allowing me to continue to do this exciting research. So, thank you.